Hello, in this video we're going to be talking about chromosomes and their role within cell division. So let's not waste any of your time, let's get right to it. Now when we're talking about chromosomes in cell division, one of the things that we're going to emphasize in this video is the fact that although at each stage, and stage in air quotes, at each stage that the DNA may find itself in, it's the same material. The difference is its level of compaction, its level of organization, and we're going to be talking about the proteins that are a part of each of those processes. Excuse me. But before we get into that, let's talk about chromosome sets, chromosome pairings. What that diagram beside me there, that's a karyotype. To the far right, when they're just loose, then the small box in the bottom left-hand corner, that's the homologous chromosomes paired together. Now, if you think about where those chromosomes come from, in a human standpoint, each organism will have different numbers, but for us, we have a total of 46 chromosomes in each cell, 23 pairs. Remember, one comes from the mother, one comes from the father. So, for example, the gene that determines the traits for hair, you'll get one from your mom, one from the dad, and that's where the pairs come from. So when it comes to, and of course what comes, when we say comes from the mom, comes from the dad, those are the gametes, that's the egg cells, the sperm cells, that kind of stuff. But if it's a somatic cell or a body cell, just your typical cell would be in the tissue of some kind. That's going to have 46 chromosomes, and that's considered diploid, which means it has both sets. So it's you'll see a 2N has the set from the mom, set from the dad. And if it's a human sex cell, the gametes, the egg cell or the sperm cell, that will only have 23 chromosomes. And that's considered haploid, 1N or just N, meaning it just has one set of the chromosomes. It doesn't have both sets. And that's where that distinction comes from. Chromosomes, when they're just compartmentalized genes along the chromosome, and the genes will encode for various proteins that will determine the traits that an organism shows. And of course, I mentioned that's called a karyotype, and diploid 2N, haploid 1N, just one set of the chromosomes. Now, when you see them paired together, they're homologous. That means they're the same size, structure, and they have the same function. The chromosome that determines height or determines hair color will match up with the appropriate one from each set from each partner, each parent, and it's homologous, as I said. Like, for example, number 3 up there, the blue ones, the third one from the left, from the top row, you can see they're the same size and shape, and they're going to code for the same traits from each of the parents. Same with 18. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 in the second row. So the fifth one, it's that white pair, it looks like, on the far right, down 3 rows. Again, same size, same shape, and they're going to have the same function between the two. Now, when we talk about, we're getting ready to start talking about the organization of chromosomes, and just like DNA, just like when we looked at chapter 3, the macromolecules of protein organization, these things can be very, very long. I mean, if you can take a DNA strand and just lay it out after it's unfolded, and it can be 6 feet, 2 meters long. That's pretty long. If you can imagine, if you're going on a trip and you're gonna pack some clothes, but the shirt that you want to take is really long, about 50 feet long, comparatively. You're really gonna have to fold that thing up to make it fit into the suitcase, or in the DNA standpoint, the nucleus. And the fact that then there's 46 of them really makes the packing, the organization, super, super important. But we're going to talk about how that process happens. But chromosomes, they're the main material is called chromatin. And of course, when that chromatin is compressed down, the chromatin is just relaxed, like maybe it's being duplicated or being prepared for duplication. Then it's called chromatin. When it's super compacted, it's chromosomes. And of course, we'll talk about when there's the two pairs, the sister chromatids, that kind of stuff. But it's the idea of the proteins that. And you'll see pictures of it. We'll talk about it. But it's the proteins that really organize it and fold it and twist it and turn it and make sure it's as compact as possible. Excuse me. Now here's the various stages of chromatin organization, chromosomes organization. It's got the 5 steps, as you can see there. The first one, just the idea of DNA being, as I mentioned, just chromatin. Maybe it's being prepared to be duplicated, or just has been. It's just stretched out. It's relaxed. The proteins have been removed, so the workers can come along and do their part. Then you got your nucleosome, solenoid, and chromatin loops, and then finally the chromosomes. And we'll talk about each one of those and the steps that go into that. Now, as I mentioned, when it's just relaxed DNA and it's chromatin, then the next step, when we start to repackage it to get it back into the nucleus in preparation for cell division, the histone proteins are added to the chromatin. And these are proteins that are just placed along the DNA strand. Again, they're going to be doing basically some wraparound folding. If you take a DNA strand and lay it out, and then you put a histone protein there, you can coil that around it, much like putting a hose away. You can fold at that point. You can do a lot of things to really, you could alternate, fold it over and fold it under, then fold it over, fold it under. You can do all kinds of things around those proteins. But what makes them super important, because they bring in a term from, again, chapter 3, with the water, the polar molecule. The DNA, as we know, in that phosphate head has that negative oxygen, which gives it a negative charge, and the histone proteins are positively charged. So there's that strong attraction, the opposites, just like in a hydrogen bond, when the polar head attaches to a hydrogen of another molecule. Excuse me. So it's the idea of that connection, getting that connection point. Now with the nucleosomes, again, we're going to coil those around the histone proteins, and when that happens, then it's called a solenoid. And solenoids are just a higher structured chromatin package. But again, the whole process is designed to really pack it in so we can get it back into, and the other 45 chromosomes into a human cell. And you can see the unit there for the weight. That's nanometers. That's a really small unit. It's hard to even compare that size, but we know that super cells are microscopic, so things inside of cells would be even more small. But the idea of during mitosis, when all this stuff is going on with the cell in preparation of dividing, the chromatins in the solenoid form, and then they're going to scaffold around or be built around proteins to really with the idea of getting that compaction as small and compact as possible. And here's a diagram that kind of just shows the steps there. And I think you can read for note taking the diagram, the text in the left hand side of the diagram. But you've got your naked DNA. Then we wrap those histone proteins in there, nucleosome, and then the nucleosome goes around more proteins, and then we're in the solenoid, and then in the chromatin loop, as you can see there. The idea of taking it and now stringing it around where it really compacts it, and then we're going to take all that and smush it down to make that form. So really the legs and the arms, if you want to mention them that way, of a chromosome really are just those chromatin loops around those protein rods in the middle. But it's just the idea of really getting it compact, really getting it so it's definitely three-dimensional, as you can see, length, width, depth, and then compacted as possible. All right. So again, this is most of the things have already been said, but the idea of when it's going through replication, single DNA molecule, chromatin, chromosomes or thread-like structures, DNA and the proteins that are going to hold them together. And once the replication has occurred, you've got the two identical DNA molecule, and that's all based on process we looked at chapter 3 with the idea of that duplication and the A, T, C, and G, complementary base pairs, and when it gets carried into RNA, of course, we bring the Y or the U in. But this whole process is designed to just get it compact and compact and compact as possible. Well, that's our video over chromosomes. When we come back together next time, we're going to be talking about the cell cycle. So we'll see you then.